a moment. So it's a moment of relief to just hear about science. I'm really excited that to hear tonight's speaker. And I was looking at his list of publications, and he has a paper called, I would have loved to have used this title, Sex and Memory, What Yeast Can Teach Us. So really looking forward to hearing more about sex and memory and what yeast can teach us. And thanks very much for accepting our invitation. And don't forget everyone to come back in 2 weeks' time for the next one. And now Mathilde.
Yes. So hello everyone, and welcome to the 7th seminar series edition. So first of all, I would like to make a quick recap of the rules to be sure this seminar goes well. So please keep your mics and your video off for a better fluidity. Only our speaker would have his video and mics on. Our speaker will share his screen. So to enjoy this seminar, please use the speaker view or affichage intervenant on the top right of your screen so that you can see our speaker's screen and video. And if you have any question, you can write them at the end of the presentation in the public chat. So I will read out loud your questions at the end of the presentation. So please, no private message to our speaker. And now let me introduce our speaker. And it's our pleasure to welcome Dr. Fabrice Caudron. He is a researcher working on cellular memory and cellular aging process at Queen Mary University of London. And today the subject is memory the bright side of cellular aging? So it sounds really interesting, and I will let him start.
Thank you very much for your invitation again. I will try to share my screen. Okay. All right. So it's a great pleasure to be invited by students. That's the best reward we can get. So I'm very happy to be here, and I tried to stay on time. So we'll see how it goes. It's a first for me to give a seminar from abroad, so we'll see. You'll find here my contact details if you want more information later on. I'll put them at the end also, so you can always contact me. So is memory the bright side of cellular aging? We'll try to touch this question maybe throughout my presentation. What you can see here are yeast cells, and that's really what we use in the lab mostly to try to answer this type of nearly philosophical question. So the first thing I would like to do is to thank the member of my lab, which are listed here. Some of them are on the picture, some of them you may know, and past collaborators also at ETH, where I did my postdoc with Yves Barral, and Julia and Marek were working with me. Juha was also at ETH, is now a professor at Helsinki University, and 2 collaborators from Stanford University. We're also funded by Queen Mary and the BSRC, which is the equivalent of the French Minister for Research. So I guess we can all agree that we can all relate to aging very easily because there are many aspects that we see from when we're born, when we grow, and then we get old and eventually die. That's something that happens to everyone, to most organisms. And really the question is, what is aging and what is driving aging? So in this scheme, you see curves that we call survival curves. And what we see, if we look at nowadays curves, is that human death is more likely to happen when you're old. Everybody can survive most of the time, fairly long, but then at some point, there's a big increase in mortality. We see it nowadays very well, but in the past, when people were dying of diseases or accidents, this was not as obvious. But now we see that really this is age-related diseases that kill us. And one aspect that we often overlook a bit is that our offspring is not born old. these, luckily, our babies do not inherit our age, and they are born young and rejuvenated. And this is really a question we want to understand. So how is this working? And there is a simple explanation for many, or not a simple, but there's an explanation for many organisms, which is that the germ line that gives the gametes is separated from the soma, from the body, and that's a simple way to rejuvenate and to have a specific germline that will form the new babies for us. But even at the cellular level, we see that the offspring can be born young. And so in the lab, we use Saccharomyces cerevisiae, which is budding yeast, or baker's yeast, or brewer's yeast. So we can do a lot of things with it. And if we look at single cells of budding yeast, we can see that they also age. So meaning that they will have a limited division potential. So they can only divide a certain amount of time. So here what you see is a cell that is dividing. It's trapped by this pillar. It's a PDMS pillar, so it's a soft material. It's trapped, and there's always medium that is flowing. It's in a microfluidic chamber. And you see that this cell comes and produces daughter cells and daughter cells and daughter cells that are flushed away, so they disappear from the movie. But this cell will divide a certain amount of time, and then you see it's slowing down. It's acquiring wrinkles. The vacuole, which is the lysosome inside the cell, starts to be bigger, and the cells become slower, and then it will die. And if we look at many cells, what we see is that we have a survival curve that is very similar to what happens for humans. So we have cells that are alive at the beginning, and at some point, around 25 division, most of the cells will die. But if we look at the daughter cells of a mother cell that has been aging, so we take this cell here, and then we do an aging experiment again, well, this cell can divide as many times as the mother cell, so it has the same lifespan. So the budding yeast cells are born young, as well as their mother were born young. And what this means is that there is some way of rejuvenation. So whatever has driven this cell to age is not given to the daughter cell. So these are some of the questions we really want to address. So what is inducing cellular aging at the molecular level? What are the mechanisms that allow for the rejuvenation of the daughter cells? And in this process, we started to ask, could aging be advantageous? Is there any reason why these cells are aging, and could it be advantageous? So to try to understand this a bit more, I would like to today present you some data on understanding that asymmetric cell division is a key mechanism if we want to understand cellular rejuvenation. So this is a budding yeast that is dividing. This is the mother cell on the left side, and it produces what we call a bud, or the future daughter cell. And there is one important region, which is here, at the limit between the mother and the bud. That's what we call the bud neck. And so during aging, this cell is going to divide very asymmetrically. You've seen the bud is growing while the mother cell is not growing so much. And we have identified, or scientists have identified in the past, many aging factors. These include, for example, that the vacuole, which is the lysosome, has a control of its internal pH that is different. Or, for example, that mitochondria start to fragment and become non-functional. So these are events that we can see in human cells also. What I'd like to focus on is on aggregated proteins or carbonylated proteins, damaged proteins. So this is a work that has been developed by the group of Thomas Nystrom initially, where they identified that during aging, carbonylated proteins that we see here, or damaged proteins, accumulate in the mother cell, while the bud, the future daughter cell, doesn't accumulate so many of them. So there is an asymmetric segregation of carbonylated or damaged protein. And we can see them with this protein, HSP104. I'm not going to go into the details of what this is. This is part of the chaperoning system that refolds or help degrading proteins. So there is an asymmetric segregation of this carbonylated protein. So we looked at it a bit more in details more recently, and we started to look at how does HSP104 localize during aging. So this at the top is a young cell, and we have tagged HSP104 with a GFP, and it is diffused within the cell. Now, if we look further during aging, so if we find cells which have divided many times already, and we can know how many times a cell has divided by counting the bud scar. So after every division, the cell has a scar of its past division, and we just have to count how many of these scars are there, and then we can know the age. So we know which cell is old or young. And what we see very early on is that HSP1 forms this foci, or this focus, there is only one here, that accumulate in the mother cell. Okay. If we look at it a bit more dynamically, what we see is that this focus is moving around the mother cell. So this is an old wild type mother cell, but it's not going into the daughter cell, very rarely. And so basically, here we wanted to ask, once we identified that this aging factor was present in the mother cell, how is it retained? How is it not going to the daughter cell? So we know these are aging factors because if we have ways to genetically remove them, the cells live longer, they can divide many more times. And if we induce them early on, we can, the cells will live shorter. So to try to understand how this is retained, I have to introduce just a key concept that we've been following in the lab, which is to understand the diffusion barriers as a mechanism to maintain asymmetry. So what you see here is a schematic of a yeast cell. So this is the nucleus. In yeast, the nucleus doesn't, the nuclear membrane doesn't break down during mitosis. It's a closed mitosis. So this would be the mitotic spindle. And here you have the bud at the top and the mother cell at the bottom, and here the bud neck. In green, you see the endoplasmic reticulum, and you see it's continuous between the mother and the bud. So it's one organelle that is linked. And if we look at any protein within the ER lumen inside the endoplasmic reticulum, it's free to diffuse. So we have ways to measure that I'm skipping today for the sake of time, but we can see that they diffuse freely from the mum to the bud or from the bud to the mum. Now, anything that is attached to the membrane, a transmembrane protein in the ER, will be diffusing freely within the mother compartment, within the bud compartment, but it will have a hard time to cross the bud neck. It's because here there's a specialized membrane that we call the diffusion barrier, and that is restricting the diffusion between the mother to the bud and from the bud to the mother. So because we can measure the diffusion barrier, we have identified a set of mutants that we can use in which we know the barrier is destroyed. Now things can diffuse freely between the mom and the mother. And so what happens when we disrupt the barrier? What we see is that this is a .6 delete cell. So it's a mutant cell that has no diffusion barrier. It's an old cell. And you see the HSP104 focus, the aging factor here. We don't see it going to the daughter cell, but I'm going to stop the movie. If I find the right frames. You may see here, particularly, that the daughter cells that come from this have formed a focus very early on. So what this means is that the barrier is confining not the deposit itself, but the seeds, what is nucleating these protein deposits to the aging mother cell. So diffusion barriers are therefore very important to form at the border between the mother cell and the future daughter cell, to form a block to the aging factor. So during aging, what we see is that the proteins, some proteins are collected at a single protein deposit site. What is inside this deposit site, we don't really know. We have some ideas of what is in there, but it's probably things that are damaged. And the seeds of these deposits are confined to the mother cell by this diffusion barrier. Okay, so really this is helping the cell to keep the aging factors to the mother cell and to free the daughter cells of age, basically. But really, so this was one of the aging factor, but what we wanted to go a bit further is to try to understand whether aging could be advantageous. And particularly when we think of single cells like yeast, which are growing as colonies, there are many, many cells around, and there is not a lot of cells that are very old in these populations. But could there be an advantage? So what we thought about at the time was maybe aging could be a way to keep memory of the past events. So if you imagine a yeast cell that is growing maybe on a fruit outside, it may go into the sun or the shadow, there may be rain coming. So there's lots of events that will change the temperature, that will change the salinity around. So there's lots of stresses that can come and go. And maybe there's an advantage to try to remember these sort of stresses to be prepared for the next ones. So we thought maybe memory could be the advantage of aging. And we found the case of memory, which is ideal here. That happens during sexual reproduction of yeast. So how does yeast reproduce sexually? There's two types of haploid yeast cells. They come as A or alpha. When they meet each other, so if you have an A and an alpha close together, they only want to do one thing, is to mate and to become a diploid. So the way to do that is that they send pheromone to each other, these blue and circles and red squares. So the alpha cells sends alpha factor and the A cells send the A factor. They receive these signals by receptor, and then there's a signaling cascade that stops the cell cycle in a G1 phase of the cell, of the cycle. And the reason is that there's only one copy of the genome in each cell. Then what happens is that these cells are going to grow towards each other. We have a term in yeast, we say they shmoo, and they shmoo because they take this shape of this. It's a comic book from the US from some time ago now that we don't really read anymore, but that's the shmoo, and they take a similar shape that we call the shmoo. And so they have to grow towards each other because yeast are not able to move, they are not motile, they don't have any ways to crawl, so they have to grow towards each other. And once they find each other, then they can shmoo, form a zygote, fuse and form a zygote. So you see what's happening here. Okay, so they fuse and they become zygote. Now this cell is diploid and it can reproduce. That's the opposite here. This cell, there's only a mat A in this case, and it's just dividing. Okay, there's two states, they can divide, bud, or they can arrest and shmoo. So what we've been doing in this context is a bit tricky. We remove the alpha cells and we only give the pheromone to the A cells. So the question is basically, what if there is pheromone but no mating partner? So here you have an A cell, the alpha cell is behind this door, sending the pheromone, but we're not going to open the door. So what happens? We place the cells in a microfluidics chamber, and then we flow in pheromone. And what you see, it's going to loop. What you see is that the first cell is going to shmoo, 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 and then at some point, it stops shmooing, and it decides to bud, and bud again. So there is pheromone in the medium, we'll come back to that, but you see it's budding, 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 budding. So once it has decided to stop shmooing, this cell doesn't want to shmoo again. It's just buds, and at the next cell cycle, it could decide to shmoo or bud, but no, it's over. It doesn't want to shmoo again. So the yeast cells can escape permanently a pheromone-induced arrest. And that takes the form of a memory because we can remove the pheromone, add it back, and the cells can keep their memory. And they keep this memory for as long as they live. So they will die, they will never shmoo again. We can quantify this. So if we look at just the blue solid curve, at the beginning, all the cells are schmooing, and with time, they escape the pheromone, so they start budding again. And then most of the cells within 16 hours will be able to escape pheromone arrest. Okay, we don't care about the other curves here. So what is the mechanism for cells to escape pheromone arrest? Well, it involves a protein, that's our favorite protein, which is called WEE3, and WEE3 is a clean-3 inhibitor. So WEE3 is this protein here. It has several features. One of them is that it can bind mRNAs. And one of the mRNA, the most studied for the case of WEE3, that it binds is clean-3. So clean-3 is a cyclin, it's a G1 cyclin, one of these proteins that can bind to CDC28, which is called also CDK1 in human cells. So it's a cyclin-dependent kinase. So it's a main driver of the cell cycle. And so this CDK1, together with this G1 cyclin, is triggering the G1 to S phase transition, okay, by activating genes. So WEE3, what it does normally is that it's binding the clean-3 mRNA, and it's inhibiting its translation. At some point, when the cell reaches a size that is big enough for the cells to enter mitosis, what happens is that clean-3 inhibition is released. WEE3 releases clean-3 mRNA. It can be translated. Then there is clean-3 protein that can activate CDK1, and then they can enter the cell cycle. Okay, so if we remove WEE3, what happens is that the cells, so this is the wild type, escape. If we remove WEE3, what happens is that the cell escape very quickly. And that's because clean-3 now is activated prematurely. So now the cells can escape very quickly. So we need clean-3 activation. We need WEE3 inhibition to delay the escape from pheromone arrest. If we delete clean-3, what happens is that the cells cannot escape pheromone arrest, showing that really you require clean-3 activation. And if we use a double mutant, clean-3 delete, WEE3 delete, then the cells escape as a clean-3 delete single mutant. So that tells us that WEE3 is upstream of clean-3. But how is it working? So what this told us is that probably WEE3 is involved in the escape. But WEE3 has another property. It has this RNA binding domain, but it also has a polyglutamine and a polyasparagine domain. So these are prion-like domains. They have different features. So it's not only glutamines and asparagine. There are more bias in these domains in terms of amino acid composition. What they are is that prion-like domains are involved in allowing conformational changes. So proteins can take different folds. And are usually self-templating. So once a protein has changed its fold, just by touching the other protein of the same nature, it can change its fold again. And this change of conformation is associated with a functional switch. So the most well-known prion that you would know is the one from the Creutzfeldt, the mad cow disease. But there are many prions in yeast. And this functional switch that prions can adopt is self-perpetuating. So it means that it can self-maintain without any input from any pathways or any energy. And so that is an interesting property if you want to maintain a cellular memory. So you can switch the function of a protein that is long-lasting. And what do we see? If we look at WEE3, normally, without pheromone treatment, it forms granules and it's distributed throughout the cell. Now, after prolonged pheromone arrest, what we see is that it forms what we call super-assemblies, which are localized here. And these super-assemblies depend on the prion-like domain. So we have a model for this type of cellular memory in which WEE3 normally is inhibiting clean-3, and that allows the cells to shmoo. That prevents the G1S transition. After some times in pheromone, and for reasons that we don't really know exactly at the moment, but that's some of the projects we have in the lab, is that WEE3 changes conformation, and we know it biochemically. This releases the inhibition clean-3, which can now activate CDK1 and the G1S transition. Okay. So can we retest this? does it really need the conformational change of V3? Well, that's the power of budding yeast. We can do mutations, targeted mutation within the genome, and we've replaced either the poly Q or the poly N of V3 to try to prevent it changing conformation and see what happens. So that's what is happening. Here we've replaced one of the two prion-like domain. And what you see is that now the cells are schmooing, and they never realize that there is no mating partner. And yet at the end, if you look at here, when the cells are looking very weird, it's because they are dead. But there is another one that is interesting, is the cell in the middle here. It's schmooing, and then up it escapes, it forms a bud, but then it forgets, and then it schmoos again. So these prion-like domains are important for two reasons. First, they can establish the escape, but they also are required to maintain it. Otherwise, these cells can learn, but then they forget straight away. But what about the daughter cells? So I've been talking about aging at the beginning. I'd like to come back to that. If we look at the daughter cells from the first movie, if you remember, what happens is that they all schmoo. They are never budding when they are born. So they don't inherit the memory from their mom. And if we look at V3 super-assemblies, we see them staying in the mother cell and not going to the daughter cell. So the assemblies are confined to the mother cell. And so that brought us to give a new name to this type of protein. On one hand, we have prions. There's quite a few prions that have been identified in budding yeast, but in many organisms, and more are identified in many organisms to have functional roles, not only to cause diseases. So prions have this change of shape that is self-templating, but they have a very important definition in budding yeast, or in animals also, is that they are infectious, or they can spread from the mom to the daughter. On the other side, we have nemens. That is the case of V3, which is probably self-perpetuating also, self-templating, but this is confined in the mother cell. And that's a very important difference. So prions are based on a self-templating conformational change that spreads to daughter cells, while nemens are very similar, but they are controlled. There are ways to retain them in the mother cell. How is it working? So I'm coming back to this diffusion barrier that I've introduced at the beginning, and are they involved at all in this case? So here what you see is another mutant. It's a BUD1 delete, but we see the same with BUD6 delete or other mutants. What you see is that the cells are schmooing normally and then they escape. But look at the first daughter cell. When it's born, it's budding, 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 budding. So now the daughter cells, and if you look at the second one, it will come here. It's born and it buds. So these cells are now budding when they are born. So they transmit the memory to their daughter cell. And indeed, if we look at these super-assemblies that we see here in the mother cell, and not in the daughter cell, in the wild type, in any of these mutants which don't have a diffusion barrier, we see the super-assemblies in the mom, but also in the bud. In the mom, but also in the bud. So really you need V3 to go into the bud, because we've opened the barrier, to give this memory to the daughter cells. So different barriers confine the V3 nemen to the mother cell. So why do they confine it? It's because V3 associates usually very closely with the ER membrane. So this is V3 that is tied with neon green. It's like a GFP. And here what we see is Sec61, which is a marker for the endoplasmic reticulum. So you see the cortical endoplasmic reticulum, and this is the nuclear membrane. And Sec61 is part of the translocon, which is a very important protein for the ER. And what we see is that super-assemblies are very often, or most of the time, if not always, opposed close to the ER membrane. Now, if we look at the prion, and the most well-known is sub-35, what happens here is that we see that the dots here are the prion form of this protein. They are often away from the ER membrane. Okay, so a very important difference between the prion and nemens is that the nemons are associated with the ER membranes, while the prions are not. And because they are associated to the ER membrane, they can be retained by the diffusion barrier. They can be controlled. So what's the deal with aging? Coming back to what we see, what I was alluding at the beginning. An important phenotype of budding yeast is that in old cells, there is no mating happening. We looked what this was due to WIS3. This is a young cell with WIS3 that is very even around the cell, but in old cells, WIS3 aggregates. And this is linked to the fact that they cannot mate. So many old cells do not, they are budding when we give them pheromone, while young cells are responding, they are not budding. Now, if we prevent the aggregation of WIS3 by removing the prion-like domain, these cells can shmoo again, okay? And really like before, there was, you can see that this cell, that is a very old cell that is budding, you see all the daughter cells here are shmooing. So this asymmetry is again dependent on the diffusion barrier. But what is interesting here is that when we tested, then we tested whether this had any impact on aging. And if you here see a wild type that is aging, this is a cell that cannot aggregate WIS3. We have prevented the aggregation of one protein, and these cells can live longer. So having the ability to age, to form memories, sorry, is a cost and restricts the lifespan. So diffusion barriers allow the retention of aging factors in the mother cell, and therefore daughter cells are born young. Asymmetric cell division and aging also allow for the appearance of cellular memories that are inherited only by the cells that have experienced the stress and not the whole colony. And that allows the cells to be naive and maybe to adapt to other stresses. In the case of pheromone response, the memory is encoded by a nemon, which is a new case of protein with a conformational change that is self-perpetuating, but it's confined. But as a cost to it is that this conformational flexibility may be, the cost may be that you age faster. Whether we age faster because we are able to memorize is an open question. And finally, I would like to say that nemons are different with prion because by their association with internal membranes, so whether you target a protein to a membrane and then subject it to restricted diffusion by the barriers has an important implication. But are nemons and prions so different? I just put a final slide to show you that if we open the barriers, very unlikely, but it happens, then WIS3 can transform into a prion, and it forms this, the cells can form this colony. So you see most of the cells are shmooing, and they are arrested completely because there's a really high concentration of pheromone here. But you see that sometimes colonies can form, and this is due to a prion form of WIS3. All right, so with this is our model. WIS3 changes conformation to allow memory. That's linked to membranes. We don't know what this green thing is, but it doesn't go to the bud because WIS3, because there are diffusion barrier at the bud neck, and WIS3 is functional into the bud, into the future daughter cell. On the other side, we have prions, which can diffuse freely and are infective. And the last movie I showed you was not demonstrating, we have many more data to support it, but showing that WIS3 can transform into a prion and then diffuse to the daughter cells. Thank you very much for your attention. Again, you can contact me here, and I'm very happy to take questions.
So thank you for your very interesting presentation. And now it's time for questions. So you can ask them in the chat and I'm going to read them. So first question from Jonathan. How many aging factors are there that are retained in the mother cell?
Sorry, how many aging factors?
Aging factors, yes.
There are many. Actually, all of the ones we know are retained into the mother cell. And this goes from DNA molecules to proteins to organelles. So mitochondria, all mitochondria or aggregated mitochondria are retained into the mother cell as a way to triage them and to selectively stop them.
Okay, thank you. Another question from Jonathan. What happens if we perturb the retention and the aging factors can leak into the daughter cell?
So what happens? What happens is that when we open the barriers and then the aging factors can go to the daughter cells, what happens is that suddenly the mother cells can live longer because they get rid of the damage.
Thank you. So another question. Do mammalian cells have a similar asymmetric retention of aging factors or mnemons?
Okay, so this is quite new. So we don't know if there are mnemons really in mammalian cells, but the diffusion barriers, and that's something I've skipped, they are absolutely conserved in mammalian cells. They are found at many places, at the base of dendritic spines, in dividing cells, and in many other instances actually. So yes, this is probably conserved.
Thank you. Another question from Gaël Fortin. He said, great talk. He said, you said that age-induced proteins deposits are retained in the mother cell. Is that also the fact in bacteria? Is the mechanisms based on diffusion barrier shared with bacteria?
Not as far as I know. One important thing in bacteria, there's no internal membrane. So that would be different. And there are other ways of probably having these mechanisms. So bacteria do also show signs of aging, and there have been reports showing asymmetric segregation of protein aggregates or other types of potentially aging factors. And so the mechanism there may be different. They may rely on one important thing that I didn't go over too much, is that these protein deposits, they don't form many, they form mostly one. And to have only one protein deposit is already an asymmetry because it can only go to one of the two daughter cells. So that may be the way bacteria do it.
Thank you. Another question from Constance Ciodo. Does Y3 bind other mRNA?
Yes, so it does bind a lot of mRNAs. And that's something we would like to try to understand a bit better. So there are, for budding yeast, the most well studied is Clean3, and that's why and for us that was the most important. It may have other impacts during aging when Y3 start to aggregate, and that's an important question.
Okay, thank you. Another question from Constance. Did you do a delta RRM Y3 mutant, and does it behave like a delta Y3?
Yes, so if we remove the RNA binding domain of Y3, what happens is that Clean3 mRNA is released and it behaves exactly like a Y3 did it. Yes.
Thank you. Question from Louis Marstrand-Dossé. Sorry. Your study concentrates on yeast cells. Do you know if there are any examples of roughly the same process in other organisms?
So in terms of asymmetric cell division and different barrier, yes, there is beautiful work from the lab of Sebastian Jesperger demonstrating different barriers during aging, that the barriers are also going down during aging, so they are down-regulated, and that leads to less asymmetry of asymmetric cell division, basically. In terms of mnemons, we know many proteins, in principle, have everything in place to behave as a mnemon. Now, this is really research to be done. It's an open question completely.
Thank you. Another question. Is there any evidence for the role of the ER diffusion barrier in asymmetric cell division in mammalian cells?
So what we know is that there are yes, diffusion barriers in cell division in mammalian cells, and this is happening for mice and rodents in general, rats and mice, essentially.
Thank you. A question from Yanis Belosif. Do you know how are acting molecularly the aging factors within the mother cells? Are they implicated in the death of the cell directly, or are there only some kind of biomarkers of all cells?
Okay, that's a very difficult question. Okay, so how do we define an aging factor? Essentially, what we would expect is that if we have more of it, it would induce death more rapidly. If we remove or if we can prevent its formation, the cells should live shorter. And so there are many molecules, if you wish, that behave like this. Exactly how they work is there's many different things coming together that will stop the cell cycle, for example, that will stop proteostasis and lead to death. So this is still an active field, and there's no definitive answer for yeast.
Thank you. Another question from Jonathan. Are there any use factors that are asymmetrically segregated only in the baby cell?
There is no use factor as far as we know. There are factors that are specific to the daughter cell, but that we don't know if they could act as use factors.
Thank you. Are there any more questions? A question from Constance. Does CLN3 essential in yeast, and can you delete it?
Yes, we can delete CLN3, so that's completely fine. The cells are not completely happy. They are very big because they spend a long time in G1 and they grow before entering the cell cycle. But yes, we've been using them a lot.
Thank you. Other questions? Okay. So I think there is no more questions. So thank you for this great presentation, and thank you all of you for your active participation. So the video of this seminar will be soon available on the G.E.N.E website, and you will receive by email a survey about this seminar. And if you can take the time to answer it, it would be nice. And so thank you. And if you can please upload with a reaction or saying thank you in the chat, it would be nice.
Thank you very much.